We are Shosei Imai and Saaya Hario from the Campbell Lab at the University of Tokyo. And today we will present single fluorescent protein-based biosensors for visualizing metabolic dynamics. And thanks again for organizers for giving us this great opportunity to share my research. So here is a link to the preprint we have published recently, last year. And this is the QR code to the adogen and plasmids uh, described in this research are available from this QR code. So please check them if you are interested. So first, I will explain the design of the biosensors we are working on, since there are several types of biosensors. These biosensors consist of a fluorescent protein and the binding protein, and they are sometimes called single FP-based biosensors. The binding protein changes its conformation upon binding to the target molecule or ion, and it alters the microenvironment of the fluorescent protein's chromophore, shown here, and it causes a change in the fluorescent intensity. They are mainly used for live cell imaging, and because they are genetically encoded, they offer advantages such as low invasiveness and high spatial-temporal resolution. As a result, many biosensors have been developed for a wide range of targets. For example, calcium sensors such as Gecko series and GCAMP series have revolutionized cell biology and neuroscience. Then, how do these biosensors change their fluorescence intensity? Here I will explain two key concepts. The first one is the pKa shift. This is the chemical structure of the GFP chromophore, and it has a phenol ring here derived from tyrosine residue. When this phenol group is protonated, it means, OH form. GFP absorbs light at around 400 nanometer and emits very little fluorescence. When this phenol ring group is deprotonated, like this, O minus form, GFP absorbs longer wavelength light at around 490 nanometer and emits strong fluorescence at around 510 nanometer. Most biosensors rely on this mechanism, that is, the pKa changes upon binding to the target ion or molecule, and it increases the population of this bright form, and thus the fluorescent intensity increases. The next key concept is circular permutation. This is a zoomed-in structure of GFP, and this is the phenol link. And this phenol link points towards the seventh strand of the beta barrel, and these orange and purple residues closely interact with the chromophore. To make GFP more sensitive to the outer environmental changes, we connect its original N and C termini with a short peptide linker and create new termini at this position. This procedure is called circular permutation and makes GFP more responsive to conformational changes because GFP chromophore is more exposed to the outer environment. Then we insert this circularly permuted fluorescent protein or CPFP into the binding protein. Okay, then next I will move on to the main part of the preprint. Although we don't not have time to present all details today, our lab, Campbell Lab, has worked to visualize metabolism as a whole by developing metabolite biosensors such as steroid and lactate biosensors. As the next target, we focus on pyruvate. As you may know, pyruvate is central to energy metabolism, connecting glycolysis and TCA cycle. Therefore, visualizing real-time changes in pyruvate concentration is highly important, but high-performance pyruvate biosensors were not available. In this research, we developed green and red pyruvate biosensors. Here is our development strategy. As shown earlier, CPFPs, circularly permuted fluorescent proteins, are inserted into the binding protein. For the CPFP component, we use CpGFP and CpMAPO. MAPO is a kind of derivative of our red fluorescent protein. And for the binding domain part, we use PDHR, which is a transcription factor that selectively binds to pyruvate. It is, derived from transcription factor and contains DNA binding domain and ligand binding domain, and we removed its DVD beforehand to, avoid any possible interference with the endogenous system. Prototype sensors were identified using these strategies, and but their performance was still poor at this stage. Therefore, we applied directed evolution to improve them. Directed evolution is a method of protein engineering and involves generating gene libraries and screening for the best variant. This cycle is repeated multiple times, again and again and again, to achieve the desired properties. Due to time limitations, I cannot go into the detail of the engineering process, but finally, we obtained bright and high contrast pyruvate biosensor series. These are the model structures predicted by AlphaFold 3, and the mutations accumulated during the evolution are highlighted. First, I will introduce the green Pi1 series, which are green pyruvate biosensors. After extensive engineering, we obtained a variant with a delta F over 0 of over 40. This delta F over 0 value, calculated using this equation, represents the ratio of fluorescence increase. Such a large dynamic range is especially beneficial for live cell imaging. Furthermore, we created affinity variants because intracellular pyruvate concentrations ranges from tens to hundreds of micromolar dependent on cell types or tissue types, and can be even higher in certain metabolic disorders. Therefore, by modifying the putative binding pocket of PDHR and surrounding residues, we obtain 4 variants with different affinities but similar properties. This allows users to choose a sensor that perfectly matches, their expected concentration range. The KD values spans from 35 micromolar to 2.7 millimolar, covering the entire physiologically relevant concentration range. The highest variant refers to the highest affinity with the lowest KD, and the lowest variant has, the lowest affinity with largest KD value. It's a bit confusing, but highest means highest affinity. We also report a control sensor designated as D green pi 1 that does not change percent, but has similar pH sensitivity. This control is essential because metabolic perturbations often involve pH changes, and it helps confirm that observed percent changes are not pH artifacts. We also tested selectivity and to photon excitation, and these sensors are sufficiently selective to pyruvate and compatible with two photon excitation imaging. Actually, two single FP-based green pyruvate biosensors called Green Pegasus and Pyronics SF had previously been reported, and we also characterized their performance and directly compared them with Green Pi 1. As a result, their fluorescence changes were much smaller, just delta F over 0 of around one-fold, and their selectivity was lower. In addition, only a single variant with a fixed KD value was reported for each sensor, limiting their applications. So collectively, we believe that our Green Pi 1 series can be a strong alternative. And we also developed Apple Pi 1 series, which are red pyruvate biosensors. Red biosensors enable multicolor imaging and optogenetic manipulation, and they are particularly useful for in vivo imaging because longer wavelength light penetrates deeper into tissues, causes less phototoxicity, and reduces autofluorescence. Similar to the Green Pi 1 series, we report 4 affinity variants with high delta F/0 values and control sensor as well. Another thing to note is that Apple Pi series exhibited fluorescence lifetime change as well as intensity change. Fluorescence intensity is dependent on many factors such as the expression level of the sensor protein, photobleaching, laser power, and so on. But the lifetime is an absolute value independent on the flow for concentration or equipment. Therefore, fluorescence lifetime imaging microscopy, or FLIM, is recently drawing attention as a quantitative imaging method. Although 0.7 nanosecond lifetime change is not optimal yet, this result demonstrates the potential for quantitative imaging. Okay, let's switch to Saya.
Okay. Do you see my screen?
Yeah, looks great.
Yep.
Okay, thank you very much. So, hello, this is Saaya Hario. I would like to introduce the mammalian cell imaging with the Green Pi 1 series and Apple Pi 1 series that we developed. So first, to demonstrate the utility of the pyruvate biosensor series, I expressed them in HeLa cells and characterized the responses upon exogenous pyruvate addition. So first, to reduce the endogenous pyruvate concentration, I carried out some pharmacological pretreatment with mammalian cells to stop the pyruvate production. The graphs show the time courses of delta F over F of the sensors expressed in HeLa cells with pharmacological pretreatment. The all sensors except control sensors show very high delta F over F upon the addition of 10 millimolar pyruvate. For example, Green Pi 1 high show the delta F over F of 23, and Apple Pi 1 high show the delta F over F of 8.6, while control sensor maintained negligible responses less than 0.5. The Green Pi 1 series exhibited constant fluorescence intensity over 15 minutes, while the fluorescence intensity of Apple Pi 1 series gradually decreased, probably due to photobleaching. Next, to visualize the intracellular pyruvate transport between mitochondrial and cytosol, I conducted imaging under the pharmacological treatment. I expressed the Apple Pi 1 highest, the RFP-based pyruvate biosensor in cytosol, and Green Pi 1 highest in mitochondria. Pyruvate is transported from cytosol to mitochondria through mitochondrial pyruvate carrier. MPC exists in the inner membrane of mitochondria. As a fuel of TCA cycle, and UK5099 inhibits the MPC. When the pyruvate is added first, followed by the addition of MPC inhibitor around 15 minutes, the concentration of pyruvate in both cytosol and mitochondria increased upon the pyruvate addition, and then both decreased after the addition of MPC inhibitor. But on the other hand, when the MPC inhibitor was added first, followed by the addition of pyruvate, the concentration of pyruvate in cytosol increased, while the pyruvate in mitochondria decreased upon the addition of MPC. And then the concentration of pyruvate in mitochondria did not increase much upon the following pyruvate addition. So our pyruvate biosensor series enable the multicolor imaging of pyruvate dynamics, suggesting they have properties that can visualize the pyruvate dynamics. So in the previous study, I managed to observe the L-lactate oscillations when the mammalian cells were treated with glucose after a few hours of starvations, using the GFP-based L-lactate biosensor that I developed. We established the mathematical model based on the imaging data investigating this phenomena in the previous study. To visualize the oscillations of both L-lactate and pyruvate, I co-expressed the GFP-based L-lactate biosensor ILACO 1.2 and Apple Pi 1 highest RFP-based pyruvate biosensor in cytosol and HeLa cells. So after a few hours of starvation, I added 5 millimolar of D-glucose. So I'm showing the video right now, which resulted in the oscillations from both lactate and pyruvate biosensors. The amplitude of Apple Pi 1 highest oscillation is much smaller than that of ILACO 1.2, but both oscillated at the same frequency, showing a time lag between their phases. In addition, when the lower concentration of D-glucose 500 micromolar was added, In addition to the oscillation with these high frequencies that we previously observed, the slow frequency oscillation was also observed. By applying the multiple metabolite biosensors, we will be able to further investigate the mechanism of glycolytic oscillations based on the experimental data. Another important application we tested is turning our intensiometric biosensors into emission ratiometric biosensors. So as Shosei also introduced, intensiometric biosensors show very large change in fluorescence intensity, visualizing the small change in pyruvate concentration. However, it lacks the quantitativeness since fluorescence intensity can be changed by expression level and so on. So lifetime is also the solution, but also I aim to turn them into emission ratiometric biosensors. So recently there was a report of emission ratiometric biosensor by fusing long Stokes shift M orange to existing GFP-based calcium biosensor. They shifted the excitation peak of M orange to shorter wavelengths so that it can be excited with GFP simultaneously while maintaining the emission wavelength. So this graph shows the emission spectra in different calcium concentration of LSSM orange fused to calcium biosensor, this construct. Although LSSM orange emission is constant compared to the GFP emission, LSSM orange emission is not completely independent from calcium concentration. I hypothesize that this can be improved by creating FB with even longer Stokes shift. So I created LSSM Kelly, a far-red FB that can be excited with GFP excitation. By introducing rational mutations around the COM4, LSSM Kelly 1 exhibits an excitation peak around 450 nanometer while maintaining the far-red emission. So this can be excited with GFP. So I fused this LSSM Kelly 1 to N-terminus of green pi 1. The emission spectra was obtained with purified protein showing pyruvate dependent fluorescence intensity change around 500 nanometer while emission around 650 nanometer remained constant. Although fluorescence intensity of LSSM Kelly is much smaller than that of green pi 1, in the flow cytometry experiment, I managed to obtain the pyruvate titration graph based on the ratio of green pi 1 high to LSSM Kelly 1. By applying an LSSM FB, we can turn the intensiometric biosensors into biosensors with large ratiometric change with single excitation, leveraging their high performance. So we're now expanding the palette of our FB-based metabolite biosensors, especially the metabolites involved in glycolysis. For example, we have created many types of biosensors targeting citrate, which is a major intermediate in TCA cycle. In addition to GFP-based intensiometric biosensors already reported on paper, we also created RFP-based intensiometric biosensors and GFP-based excitation ratiometric biosensors, another type of ratiometric biosensor. So we're creating the biosensor with different targets, different colors, and different types to extend the applications. Using our metabolite biosensors, together with optogenetics tools regulating the enzyme activity, we will further investigate the glycolysis. So thank you very much for your attention. Again, you can access to the preprint or plasmid available on our gene through this QR codes. So thank you very much.